Disease causation and natural history of disease. In this lecture, we are going to investigate first causation of disease. And in the next lecture, we are going to study the natural history of disease. Natural history of disease. Natural history of disease refers to the progression of a disease process in an individual over time in the absence of treatment. For example, untreated infection with HIV causes a spectrum of clinical problems beginning at the time of seroconversion. This is the primary HIV, and terminating with AIDS and usually death. It is now recognized that it may take 10 years or more for AIDS to develop after seroconversion. Because the spectrum of disease can include asymptomatic and mild cases, the cases of illness diagnosed by clinicians in the community often present only the tip of the iceberg. This is only the portion of those who have the disease that have been presented to the health services or they seeked healthcare, so we know them. There are nevertheless many additional cases that may be too early to diagnose or even may never be diagnosed because they do not progress into the clinical stage. Unfortunately, persons with inapparent or undiagnosed infections may nonetheless be able to transmit infection to others. This is very applicable to the case of COVID-19 that we are going through. The number of cases that is being reported every day in the news represents the percentage of those who have been tested for this day and proved to be positive. We only hear also about the number of deaths for this day and the number of those who have been admitted to hospitals because they have symptoms and needed health care. We also know the number of those who need ICU, they are in intensive care, or needed to be on artificial respiration. This is only the tip of the iceberg. What is really troublesome is this too many cases in the community that are having the disease but without any symptoms. They have subclinical and we may never know them. They get the disease, they get through it, they got healthy again, but they have infected so many people when they were carrying the disease. Such persons who are infectious but have subclinical disease are called carriers. Frequently, carriers are persons with incubating disease or inapparent infection. Persons with measles or hepatitis A, influenza, and several other diseases become infectious a few days before even the onset of symptoms. However, carriers may also be persons who appear to have recovered from their clinical illness but remain infectious, such as chronic carriers of hepatitis B virus or persons who never exhibited symptoms. The challenge to public health workers is that these carriers, unaware that they are infected and that they are infectious to others, they are sometimes more likely to unintentionally spread infection than other people with obvious illness. These are the dangerous group in the population. So, for example, during the current COVID-19 epidemic, you should perceive anybody in front of you that you deal with as infectious, even your own family, literally anybody, or that you may be infectious and you should protect others. Unapparent infection. Unapparent infection can be either preclinical disease or subclinical disease or chronic carriers. Preclinical disease is the early stage of disease progression. The disease is not clinically detected but is destined to become clinical disease. Subclinical disease is the case where disease is not detected, but the host carries the organism or has antibody response. Chronic carriers are those who continue to harbor a pathogen such as hepatitis B or Salmonella typhi, the causative agent of typhoid fever, for months or even years after their initial infection. All of these cases, people with preclinical, subclinical, or chronic carriers can all be infectious to others without even knowing about it. One of the very important cases about unapparent infection is called Typhoid Mary. This is one notorious carrier. She was Mary Mallon. Oh, they called her Typhoid Mary. She was an asymptomatic chronic carrier of Salmonella typhi. As a cook in New York City and New Jersey in the early 1900s, she unintentionally infected dozens of people until she was discovered, and then she was placed in isolation on an island in the East River, where she died 23 years later. This figure shows the iceberg concept of infectious disease. This is what we called the tip of the iceberg. On the right side of the figure, we can see the host response, and on the left, there is the cell level response. If we start at the bottom of this iceberg, the very bottom below, there is starting with the exposure of a susceptible host to an agent, but without infection first. If we go a step up there, the infection starts, but with subclinical illness, asymptomatic infection. On the left, there is the cell response, where viral multiplication starts, but without any visible changes on the body, or incomplete viral maturation. Here, up there, we have the C level. What presents to the health services, what we can see and have been diagnosed as cases, are only the cases who start to have the clinical infection with moderate severity or mild illness, who start to seek healthcare or have apparent symptoms that people around them can see. On the cell level, here we start to have cell transformation and cell dysfunction. If we go up the pyramid, there are the classical and severe cases, and at the very tip of the pyramid, the proportion of people who die from the infection with the microorganism. So what we see, what we report, the cases that we know about and we are counting is only this portion, the tip of the pyramid that is up the sea level, those with moderate and mild illness and severe cases who are receiving health care. This is only what we know about. So we should appreciate that beneath the sea level, down there, we have more than 5 times the amount of those who have been diagnosed and counted as cases. Most of them are having subclinical infection. They are infectious. They may never even develop the disease, but we have to be careful from them. Here we can see the distribution of clinical severity for 3 different infections. Infection with tubercle bacillus for tuberculosis, infection with measles virus, and infection with rabies virus. In the case of tubercle bacillus, unapparent infection is very frequent. Most of the cases have unapparent infection with a good proportion of very mild symptoms and a minority or very little amount of fatal cases. If we look at measles virus, clinical disease is very frequent with few deaths. We can see most of those who get measles virus will develop moderate disease and a good proportion with severe non-fatal and a proportion of fatal cases. The picture is different with rabies. Rabies infection is usually fatal in most of the cases. A person infected with rabies, a small proportion will develop severe and non-fatal cases, disease, but the majority of those who get infected, they would die if they do not receive proper treatment. Rabies is very highly fatal infection. Chain of infection. As described above, the traditional epidemiologic triad model holds that infectious diseases result from the interaction between agent, host, and environment. More specifically, what happens is that transmission occurs when the agent leaves its reservoir or host through a portal of exit, and then it is conveyed by some mode of transmission and enters through an appropriate portal of entry to infect a susceptible host. This sequence is what we call the chain of infection. Reservoir. What is a reservoir? The reservoir of an infectious agent is the habitat in which the agent normally lives, grows, and multiplies. Reservoirs include humans and animals and the environment. The reservoir may or may not be the source from which an agent is transferred to a host. For example, the reservoir of Clostridium botulinum is soil, but the source of most botulinum infections is improperly canned food containing Clostridium botulinum spores. So the canned food is the source. Human reservoir. Many common infectious diseases have human reservoirs. Diseases that are transmitted from person to person without intermediaries include the sexually transmitted diseases, measles, mumps, streptococcal infection, and many respiratory pathogens. Because humans were the only reservoir for the smallpox virus, for example, naturally occurring smallpox was eradicated after the last human case was identified and isolated. Animal reservoirs. Humans are also subject to diseases that have animal reservoirs. Many of these diseases are transmitted from animal to animal with humans as incidental hosts, if humans come into contact with these animals. The term zoonosis refers to an infectious disease that is transmissible under natural conditions from vertebrate animals to humans. Long recognized zoonotic diseases include brucellosis from cows and pigs, anthrax from sheep, plague from rodents, trichinosis from swine, swine are pigs, and rabies that can be transmitted from bats, dogs, raccoons, and other mammals. Many of the newly recognized infectious diseases in humans, including HIV/AIDS, Ebola infection, and even SARS, are thought to have emerged from animal hosts, although those hosts have not yet been identified. This is what happened also with COVID-19, where scientists are trying to understand where it came from. It could have come from bats or any other host. It is not clear yet. Environmental reservoir. Plants, soil, and water in the environment are also reservoirs for some infectious agents. Many fungal agents, such as those that cause histoplasmosis, live and multiply in the soil. Outbreaks of Legionnaire's disease that we talked about before are often traced to water supplies in cooling towers and evaporative condensers in AC systems, which are the reservoirs for the causative organism Legionella pneumophila that causes severe pneumonia. The second part of the chain of infection after the reservoir is the portal of exit. Portal of exit is the path by which a pathogen leaves its host. The portal of exit usually corresponds to the site where the pathogen is localized in the body. For example, influenza viruses and Mycobacterium tuberculosis exit the respiratory tract. Schistosomes through urine. Cholera, Vibrios through feces. Sarcoptes scabies in scabies skin lesions. Other diseases exist in the bloodstream. So some blood-borne agents can exit by crossing the placenta from mother to fetus, as in the example of rubella, syphilis, or toxoplasmosis, while other diseases or blood-borne agents exit through cuts in the skin or needles in the skin, like in the case of hepatitis B, or blood-sucking arthropods like mosquitoes in the case of transmitting malaria. Modes of transmission. An infectious agent may be transmitted from its natural reservoir to a susceptible host in different ways. It could be either direct transmission or indirect transmission. First, let's look at direct transmission. This is person to person directly. This can happen through direct contact, skin to skin contact, or through kissing, by saliva, or sexual contact, or through soil. The second way it can get transmitted is through droplet spread. This is also direct transmission. Droplets spread from a person who has the infection. Spray with relatively large, short-range aerosols produced by sneezing or coughing or even talking of a person with an infection. This is what happens in the transmission of polio, hepatitis also, HIV, and sure, influenza. The indirect mode of transmission. This could be airborne, vehicle-borne, or vector-borne. First, airborne. Infectious agents are carried by dust or droplet nuclei suspended in the air. When we say suspended in the air, this means they are very small, very lightweight particles that are less than 5 micrometers in diameter. An example for this is measles, how it transmits in a doctor's office. A child comes with measles to see the doctor. Through sneezing and coughing, measles can stay with droplet nuclei suspended in the air from this child in the doctor's office and can stay in the air for even hours. This child goes home, another child comes later and then can get the measles infection through inhaling these droplet nuclei suspended in the air that were still in the doctor's office. The vehicle-borne. Vehicle means through inanimate objects, non-living objects. This non-living object could be food, could be water that carries hepatitis A virus, could be biologic product like blood, and also fomites. Fomites, this means things like handkerchiefs, bedding, surgical scalpel, toothbrush, toys, or cutting board if contaminated food was used on this cutting board and not cleaned. These are fomites. The third way is vector-borne. Vector-borne is through small insects like mosquitoes, fleas, and ticks. This transmission could be mechanical or biological. Mechanical, when mosquitoes carry infectious agent in a mechanical way on their legs or wings. This is the mechanical way. But also there is the biologic way. This is when the mosquito has the infectious agent inside their body where it grows and multiplies. So, vector-borne could be through biological or mechanical means in the insects. Examples for this are E. coli infections, Coxsackie virus, this is hand, foot, and mouth disease, lice, meningitis, or rotavirus diarrhea. Transmission of agents from mother to child. All pathways of transmission that we have discussed earlier are considered horizontal transmission. All were horizontal. The case from mother to child is vertical transmission. We call it intergeneration transmission. This is transmission of disease-causing agents from mother directly to baby, either just before or just after birth, or via placenta or breast milk. Examples for diseases that can be transmitted from mother to baby include HIV and also hepatitis C. Portal of entry. The portal of entry refers to the manner in which a pathogen enters a susceptible host. After knowing the reservoir of the disease, then knowing the portal of exit, after that in the chain of infection, we learned about the mode of transmission, then we need to know more about portal of entry. So, it is the manner in which a pathogen enters a susceptible host. The portal of entry must provide access to tissues in which the pathogen can multiply or a toxin can act. Often, infectious agents use the same portal to enter a new host that they used to exit the source host. For example, influenza virus exits the respiratory tract of the source host and enters the respiratory tract of the new susceptible host. In contrast, many pathogens that cause gastroenteritis follow what we call fecal-oral route because they exit the source host in feces. Then they are carried on inadequately washed hands to a vehicle such as food, water, or utensils, equipment that we use for eating. And through there, it enters a new host through the mouth. Other portals of entry include skin, as an example of hookworm, enters through the skin, penetrates the skin, or mucous membrane like syphilis, or blood, as in the case of hepatitis B or HIV infection. The final ring in the chain of infection is a susceptible host. Susceptibility of a host depends on genetic factors, also can depend on specific immunity and non-specific immunity or non-specific factors. Specific immunity that results from getting infected with the disease or being vaccinated, while non-specific factors like skin or mucous membranes, they are the physical barriers against the organism. Also, there is gastric acidity or cilia in the respiratory tract or the cough reflex. We call all these non-specific factors. They all affect an individual's ability to resist infection or to limit pathogenicity of the microorganism. For example, persons with sickle cell trait seem to be partially protected from a particular type of malaria. This is an example of genetic factors. Specific immunity refers to protective antibodies that are directed against a specific agent. Such antibodies may develop in response to either infection or vaccination or getting exposed to toxoids. Factors that may increase susceptibility to infection by disrupting host defenses include cases of malnutrition, alcoholism, and disease or therapy that may impair the non-specific immune response, like chemotherapy. Implications for public health. Why is it important in epidemiology as a cornerstone of public health? Why is it important to learn about the chain of infection for any disease that we are facing? This is because knowledge of the portals of exit and entry and also modes of transmission provides basis for determining appropriate control measures. Control means preventive measures. In general, control measures are usually directed against what? It is directed against the segment in the infection chain that is most susceptible to intervention. Any place in the chain that we can block or make a break, this can stop the infection sequence. For example, some interventions are directed at the mode of transmission by isolation of someone with infection, keeping him away from other people. This way we can stop transmission, or counseling persons to avoid the specific type of contact associated with transmission, as what happens in the case of COVID-19 by advising personal hygiene and social distancing to prevent COVID-19. By social distancing, by continuous hand washing and washing surfaces where droplets of the infection can be, in this way we can break the mode of transmission. Some other strategies for control or prevention of disease rely on protecting the portals of entry. And those can be simple and highly effective, like using bed net for mosquitoes to prevent mosquito bites, also using masks, gloves, and face shield for doctor's work or dentist's work, or now with COVID-19, we are advising everybody in the community to use personal protective equipment, such as a mask or face shield, to stop the portal of entry if you face anybody who is infected. At the same time, this can also be block for the portal of exit. You don't know whether you are infected or not. Also, you can't know whether the person in front of you, either a family member or a colleague at school or a teacher or the seller in the shop, anybody could be infected. So if you are wearing your mask, everybody else is wearing the mask, so we can block here the portal of exit and portal of entry, and we can limit the possibility of transmission of the disease. Some other interventions aim to increase hosts' defense, like vaccination. Everybody is looking forward nowadays for the COVID-19 vaccine. When we'll get the vaccine? Who will get the vaccine first? What is the priority? Would it really work? Would the disease stop before the vaccine comes? Will I get the opportunity to get the vaccine? So everybody is interested in the vaccine now. Some interventions attempt to prevent a pathogen from encountering a susceptible host, as the concept of herd immunity in childhood vaccinations. When we vaccinate all children, with a large proportion of children being vaccinated, the small proportion who are not vaccinated yet most probably will not get the disease because the majority of children have been vaccinated. So, what is the concept of herd immunity, and how does it relate to disease transmission? In a population, disease transmission may stop before all susceptible individuals are infected. How? We'll see. In herd immunity, It is the resistance of a group to attack from a disease to which a large proportion of members in the community are immune to. The larger the number of people who are immune, the less likely that the other people who are not infected, that they will get the disease. This will lessen or reduce the likelihood of a person with a disease coming into contact with a susceptible individual, because the number of people in the community or population who are not susceptible because they have been infected already or because they have got the vaccination. If we look at this graph here or figure, the green dots are people who are immune. The yellow dots are people who are still susceptible. We have one person with the disease. The larger the number of dots that are green, that's the immune people, the lower the probability that this person with a disease will come into contact with someone in the population who is susceptible. The higher the proportion of immune people, the lower is the likelihood that people without immunity will get infected. How would herd immunity used for disease control? The success of herd immunity in controlling the disease depends on the proportion of subjects with immunity in a population. Immunity, of course, can be from immunization or from infection, natural or unnatural immunity. So, when the population is immunized or vaccinated at or above the herd immunity level, there is a level for the proportion of the population that should be immunized or have got the infection for herd immunity to start working. This is a critical immunization threshold level. What happens is that the infectious disease will become more rare and it will spread less and will be gradually eliminated from that population. Herd immunity level differs for various diseases according to how infectious the microorganism is. For example, it is estimated that 94% of the population must be immune before measles can be controlled. So it is very important to achieve above 94% level of measles vaccination if we want to eliminate measles among children in a population. For mumps, it is around 90%, and for polio, it is 80%. If 80% of children population have been immunized against polio, this will provide herd immunity for the other 20% who have not been immunized, and they will not get the disease. So the more infectious the disease is, the higher is the herd immunity level. There are 3 requirements for herd immunity in a population. Number 1, the disease agent should be restricted to a single host species, to be restricted among humans, within which transmission occurs. For example, smallpox only happens among humans. There is no other reservoir in the environment. Then condition number 2, there is relatively direct transmission from one member of the host species to another. Transmission happens from one person to another person, most of the time directly, with direct contact. And condition number 3 or requirement is that infections must induce solid immunity, must induce immune response a person becomes immune after being infected. If these three conditions are present, we can look at herd immunity. It can work.